Innate defenses to viral infections are triggered by pattern recognition receptors, or PRRs, present in cells of the innate immune system. During an infection, cytoplasmic PRRs RIG-I and PKR bind to viral signature RNAs, change conformation, oligomerize, and activate antiviral signaling. To monitor RIG-I and PKR conformational changes and oligomerization in vitro, human A549 cells are infected with Rift Valley fever virus clone 13 to stimulate PRR activation. Cell lysates of control and infected cells are then prepared. To analyze for RIG-I and PKR conformational changes, a limited trypsin digestion is performed. If conformational changes in protein structure have occurred, the protein is more resistant to digestion, and bands will be seen with Western blotting analysis. To analyze for formation of oligomers, native page is performed, followed by Western blotting analysis. If oligomerization has occurred, higher oligomeric RIG-I and PKR complexes are seen.
The main advantage of limited protease digestion and native page is that these techniques facilitate the direct measurement of RIG-I and PKR activation. These methods can help answer key questions in the field of innate immunity, such as what is the exact nature and origin of the RNA species?